Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 175th seminar, and I call it a Princeton University Day. I just got back to San Diego from 3 week back-to-back trips to give multiple invited talks and serve as a panelist at NSF headquarters, University of Wisconsin-Madison, and multiple institutions and a biotechnology conference in Korea. I have enjoyed discussing with so many people at NSF, Madison, and also Korea to promote international collaboration. Despite many global issues, I became more optimistic thanks to the enthusiastic researchers, especially young future leaders in synthetic biology or engineering biology, whom I met during those trips. Let's work together to solve those problems. Uh, that is what I want to see. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jose Avalos. He is a professor at Chemical and Biological Engineering Department in Princeton University, focusing on metabolic engineering research. Importantly, he recently served as the 2025 conference chair for the SIMB SIM annual meeting, which is a tremendous service for the research community. Jose, thank you so much for your scientific contribution and also the service to the community. And the virtual podium is all yours now. And thank you again.
Great. Thank you so much, Tisok, for that lovely introduction and also for the invitation to be here today. It's been a pleasure meeting you as well, Highness, and sharing this stage with you today. So today I'm going to tell you very briefly about what we're doing in my lab. If I can share my screen for a second. Is it shown properly?
Yep. Thank you.
Right. Yeah, so as you know, there's many flavors of synthetic biology. So in my lab, the flavor that we like to play most with is metabolic engineering. Particularly, so metabolic engineering, which essentially is the idea of engineering microbes to harness their metabolism to convert cheap and renewable feedstocks into valuable products that can range from biofuels, commodity chemicals, specialty chemicals, pharmaceuticals, food, etc. And so our canvas is the cellular metabolic pathway that you see here, very simplified. And so the idea is to take the cheap renewable resources, feed them into a metabolic network, and maximize its flow through the metabolic network to an output of a desired product. And to do this, you have to upregulate pathways, overexpress enzymes, sometimes some that are not endogenous to the host. You have to delete genes, etc. And the idea is to do this in the most efficient way possible while the cell's still growing. Because these microbes have been so revolutionary in sustainable biomanufacturing, they're often called cell factories or microbial cell factories. But this analogy is adept at some level, but once you look under the hood, you realize that this analogy quickly breaks down. For one thing, when you look at a true factory, you will notice that there's multiple unit operations that are encased into different reactors or modules or columns. So they're not all mixed together, right? Similarly, in the cell, you should not expect the microbial cell, the microbial factory, to behave as a bag of enzymes. So there's all these different compartments that are already there that could serve the purpose of different reactors, if you will, nanoreactors inside the cell reactor itself. And so a big part of my lab works on metabolic compartmentalization in natural organelles. This is actually work that I started when I was still a postdoc at MIT, and one of the things that I brought with me when I came to Princeton, which we really pioneered the use of mitochondria as a nanoreactor inside yeast cell to boost chemical production. And so this is the first example that we work with, in which when you compartmentalize the biosynthetic pathway for branching branch chain higher alcohols, particularly isobutanol, isopentanol, 2-methyl-1-butanol, you see a significant boost in the production. And since then, we and others have used mitochondria in many different ways to compartmentalize different pathways with very powerful results. Now, one of the things that we are doing in my lab, of course, we're still using mitochondria as a vesicle in which to contain reactions, but we're also thinking of it as the living dynamic micro organelle that it is, right? So this is an organelle that grows and divides and moves around the cell. And we're thinking of ways in which we can now use mitochondria not just as a compartment for metabolic pathways, but also as a vessel that we can engineer so that it can boost the products that the metabolic pathways that it contains. Now, this mitochondria, as you well know, is a organelle, a natural organelle that is surrounded by a biological membrane, actually two biological membranes, right? But there's another class of organelles that naturally exist in cells that have no membrane surrounding them. These are membraneless organelles, and they're also known as bimolecular condensates. And we have been harnessing them to produce synthetic organelles. And so these are, you might have heard them as P granules or signaling clusters or stress granules. The nucleolus is a classic example of a phase-separated membranous organelle. When I say phase-separated, it's because these organelles are formed by proteins that condense with each other by these weak and transient interactions of disordered regions that brings them together. Typically, they're understood, and most of the time, they're in the liquid state, kind of like an oil droplet inside of water, but they can have different forms that move into a gel state like behavior, or even solid, or even crystalline behavior. And these are sometimes related with pathogenesis. Now, we are using these as an inspiration for building these synthetic organelles. And this is work that we've done with colleagues here at Princeton, Cliff Brangwing and Jared Turcher. And so we can actually engineer these organelles by harnessing the mechanisms by which they form, which essentially is allowing them to nucleate by having a core structure, like a chassis of a protein, that is fused to disordered regions. And this causes nucleation of this protein to condense, to phase separate from the rest of the cytosol into these distinct liquid organelles, liquid membraneless organelles, that then we can functionalize with enzymes, right? So there's an example in which we functionalize a synthetic organelle with the ERK20 of the mevalonate pathway, and the terpenoid pathway, I should say, and compartmentalize it with a farnesene synthase, and then show that by doing this, you can boost the level of production of the farnesene. And then we've developed different chassis and modules so that you can control things like the concentration of the proteins that you cluster, as well as the stoichiometric ratio. We've also developed organelles that are immiscible with each other. So for example, here, by just controlling the composition of the liquid droplets, you can make them so that they are totally immiscible with each other, or maybe touching but not merging with each other. So this is how we think of spatial control, spatially controlling this microbial cell factor. Now, there's another important control that happens in real factories that we don't typically think or are able to easily engineer in the cell. And that is these closed loop controls, or one way of thinking it is a temporal control. So knowing when to perturb or when to fix your process, right? So we're familiar in our case with these bioreactors in which we have all these types of controls like temperature control or pH control, oxygen control, in which we can have a real-time readout of what is the condition. And then we have a set point, kind of like a thermostat, and then adjust when there's a perturbation or a deviation from a desired set point. These things exist not only in bioreactors, but all over a real factory. In microbial cell factories, this is really rudimentary, the kinds of controls that we have. What we've had is for decades chemical induction, which has served very well, but it has some intrinsic limitations, which is a one-step kind of addition of a chemical or consumption of a chemical. The point is that the cell grows for some time in one state, and then once it reaches an optimal state, you can add a chemical, or when the cell consumes a chemical, induce the metabolic pathway of interest so that it shifts from growth to production. More recent strategies involve like autoinduction, in which there's a genetic programs introduced into the cells so that once they reach a certain threshold, either by accumulating a metabolite or sensing the quorum of the media, then they shift from growth to production. So there's many examples in which these types of controls are very effective, right? And we've been using them for decades now, but they do have some limitations. One of them is the need for addition of chemicals, for example, and also the difficulty in reversing, going back and forth, right? So the ability to, if you overshoot, to autocorrect, right? And so to address these kinds of limitations, we have really pioneered the application of optogenetics for microbial engineering for this type of control. So because the advantage of light is that you can apply it instantly and reversibly, and it's infinitely tunable, right? You can control it by the intensity that you use to apply and the periodicity and the forcing periods that you use, the frequencies in which you apply the light. And there's many different mechanisms by which you can control cells with light. The most common one is light responsive transcription factor, so you can control transcription with light. Combinations of transcription post-translational control. So for example, when you have, you can induce optogenetically a genetically encoded inhibitor. So you're both inducing with light, say a nanobody that will inhibit an enzyme that is involved in a metabolic pathway that you only want for some part of the time. There's also post-translational controls. A nice example is one in which you have here a light responsive degron domain. So if you shine light to a protein that is fused to this type of domain, then you can degrade it with light. So that's a post-translational kind of control. And then there's also gene circuits in which you can use a combination of elements, transcriptional elements typically, but it could also be a combination of transcriptional and post-translational elements to achieve things like inversion of the response to light or amplification to the response to light. Now, the type of hardware that we use can go from very simple, right? A simple panel that you get from Amazon or from the internet, and then just shining in over your plates. Could be liquid plates, agar plates, 96-well plates. Sometimes if you're more handy with electronics, there's some of these devices that you can download online. This is work from Lucas Bugash, with whom this is an optoplate is called, in which you can illuminate a 96-well plate format, individual wells within the plate with different wavelengths of light, different frequencies, different times, different intensities. And there's ways to build, there's the blueprints to build these are online. They're not very expensive to build. And we teamed up with Lucas a couple of years ago to build an optoplate reader version of this, in which it includes a reader component. So it's the optoplate that illuminates with light, but you're also simultaneously reading optical density and fluorescence. So this is a very powerful way to screen for phenotypes and also start to work on closed loop controls. There's other types of equipments that we use. These are opto wells produced by a company called Opto Biolabs that has very refined and calibrated light properties in terms of wavelength and intensity. So this is a very reliable way to do some of the experiments that we do. Typically we use 15 mil or 50 mil scales. We can scale it up in these tiny microbioreactors of a volume about 40 mils. You can also buy these online. This is developed by Harrison Steel, but he outsourced it to a company that you can buy this for a very reasonable price. And of course, we have in-house makeshifts, adaptations to our reactors in which we surround them with LED devices and so on. So this is sort of our hardware. And we have many, many results, but I just wanted to pick one here just to illustrate that we are no longer restricted to a two-phase fermentation of growth versus production. With light, you can have any number of stages or phases, right? So you can have this reflects the amount of light that you provide. So no light, some light, full light, for example. And this is controlling the production of different chemicals of interest like lactic acid, isobutanol, and naringenin. And as you can see, you can vary these different phases by changing the duration, but also the light input that you provide. And those have profound implications in the levels of productions that you achieve. This is a small example that I wanted to show using just this type of arrangement, just to show that the threshold to entering this field is not so high. Right? So you can go through these more expensive devices, but you can start with just a panel, a $30 panel that you can buy on the internet. But when you control it in these kinds of reactors, then of course, then that's when you start to beat the titers, right, in the field. And that's when you can have these types of very fine-tuned controls. If you're interested in reading more about our work, this is a collection. I think this is a comprehensive list of the papers that have come out from my lab that do optogenetics. And there's more in the pipeline, so stay tuned. Now, there's a way that you can think of controlling both, right? Space and time. So for example, we've developed these synthetic organelles that I told you about in a way that you can control when the organelle forms. So by controlling the nucleation with light responsive proteins, then if you shine light, for in this case, you will form these compartments that will bring enzymes together just for the amount of time that you want. Vice versa, you can also do it. We also have organelles that dissipate with light. So they're in the dark, and then when you shine light, then they dissipate. Again, in this case, you're clustering enzymes in the dark and dispersing them in the light. And you can imagine if you do this with non-miscible organelles, then you can have these different compartments forming at will, right? Clustering different sets of enzymes. And we've shown that this can have an effect. So this is, for some of you might be familiar with the violacing pathway. It's a branch pathway. So we've shown that using these types of organelles, if you cluster different sets of enzymes, you will produce different kinds of products in the light versus in the dark. Another way in which you can combine space and time control in using microbial cell factories is in microbial consortia. Many of you might be familiar. The idea is that you divide a long metabolic pathway in different strains that are going to grow in consortium. And then by dividing the labor, avoiding toxicity, etc., you can enhance the production of a pathway. The challenge has always been, how do you stabilize and how do you optimize the composition of this consortium? So we've developed an optogenetic strategy in which we control the growth rate of the different members of the consortia with different wavelengths of light. So for example, you might have a bacteria that grows only in red light, another one that grows only in infrared light, another one that only grows in blue light, and another one that grows only in darkness, for example. And so we've developed these types of systems. Here is a consortium of 4 members of bacteria, growing each one in different light conditions. And so you can see this is just one example in which, depending on the amount of light that you provide, you can very precisely control the ratio of the different members of the consortium. And we've shown on sort of proof of concept studies that this can have an effect. This is actually a very simple consortia of a yeast and bacteria consortia, in which you're only controlling the growth rate of the bacterium. This for the production of 2 different chemicals, divided across the 2 strains, isobutyl acetate and naringenin. What you can see here in green is how this consortia produces in the absence of light control. In other words, non-optogenetically controlled consortia versus, this is different inoculum ratios, versus a consortia that is optogenetically controlled. So you can see very clearly that when you have control over the composition, you can significantly improve the production of a chemical produced in consortium. Now, what is the ultimate goal here? What are we aiming for. I think the dream here is to realize what we call metabolic cybergenetics. This is the idea of building kind of like a thermostat to cellular metabolism, in which you have the ability to sense what's going on with the cell, whether it's growing, whether it's there, with a proportion, if it's a consortia, whether it's metabolically active, whether it's producing using biosensors, and use this information to control the light output that you're going to use as an actuator to intervene and adjust the metabolic state of the cell to fit a desired set point that will maximize the chemical production, right? In other words, truly come closer to what's going on in a true factory. And this is the idea is not just to do this at the cellular level, it's actually at the reactor level, right? So now we're actively working on building these types of controls in real bioreactors, in which you have the ability to sense what's going on with the cell, maybe it's the level of stress of the cell or production of the cell, and then adjust how much you demand of the cell with using light accordingly. So with that, I just want to acknowledge the people that have done the work. This is the current members of my lab, former members of the lab. Key collaborators, I need to disclose potential conflict of interest. Fermit is a company that is using optogenetics for microbial fermentations and my source of funding. And with that, I thank you again, Tesok, for the invitation, and I leave the floor for Hannes, and I'll stick around in case there's any questions after his talk. Thank you.
Fantastic. When I was the graduate student, I wanted to work on what you just, described, but I never implemented except the co-localization, that is the scaffolding, idea, I published with the Jay Kitzling and also Christopher Preder, but you are already kind of implementing many of them I really wanted to achieve. So thank you so much for your contribution. That's absolutely amazing how we can use those kind of optogenetic tools for metabolic engineering application and etc. So that is amazing. Thank you.
Thank you, Tesok. It's very kind of you.
Okay. So now the main speaker of today with a longer introduction. Dr. Hannes Beyer is a junior professor and also a Zeitgeist fellow for the synthetic model biosystems at the Institute of Synthetic Biology, Heinrich-Heine-University, Düsseldorf in Germany since 2022. His current research focuses on synthetic model mice biosystems, particularly on developing optogenetic technologies for light-regulated in vitro tissue models. I think this is kind of common theme between these two speakers today. Hannes holds a diploma in biology from the University of Freiburg, where he also completed his PhD under the supervision of Professor Wilfred Weber. He is also very famous for the optogenetic tools development, I believe. His doctoral work centered on engineering plant photoreactors for optogenetic application in mammalian cells and hybrid materials. After his PhD, Hannes pursued his postdoctoral research in Helsinki, Finland, studying the biology and engineering of self-splicing proteins. That's also very interesting, another direction he has been pursuing, I believe. Hannes, thanks so much for your time today. Please take it away. It's all yours now. Thank you.
Okay, thank you. Let me share my slides. Okay, can you see it?
Yep. Thank you.
Okay, then thank you again, first of all, for inviting me to speak today. Thank you for the kind introduction to both of you. Also, your talk was fantastic and very inspiring. Thank you so much for it. And also big thanks for all the energy you put into running this seminar series. So today I would like to talk about developing synthetic model biosystems that are meant to study health and disease. And in particular, I would like to focus on the engineering of optogenetic mammalian in vitro tissues in this context. So when we talk about biomedicine in general, we focus on human subjects, but the often needed invasive methods can actually cause a shortage in sample materials. For example, when it comes to the brain, most of us would probably prefer to keep it, and this means we are then left either with post-mortem samples or we have to resort to alternative models, including animals, representing the main alternative. But there's an ongoing uncertainty as of how much situations in animals actually reflect those in humans, and there are also ethical considerations. And lastly, then there are classical cell line models and cell culture models, but even if of human origin, they have often changed in various aspects, might not adequately represent the real world, and they're also not available for each and every cell type. However, there's another form of a model system that positions itself sort of in between a 2D cell culture and an animal model, and this is what we can call a three-dimensional tissue model. And in fact, during the past years, there was quite some pressure for their development due to urgent needs. And with this pressure, they also made significant progress, in particular accelerated by induced pluripotent stem cell and cell reprogramming technologies that basically allow us to generate stem cells from somatic cells of individuals, from which we can then even now generate organ-like in vitro tissues, so-called organoids, that hold a great potential for personalized and regenerative medicine. So our aims therefore center around introducing synthetic biology and, importantly, optogenetic technologies into 2D and 3D in vitro model systems. What we would like to achieve, for instance, would be to be able to induce disease at a desired time point and also in a specific region of an in vitro tissue as it forms, which would then allow us to study disease spread or the interaction of disease tissue with healthy tissue. And this is now where optogenetic comes into play, where we basically use light signals or optical signals to quite precisely regulate biological activities. Another example where this technology would be very useful is to sculpt tissue features as needed. For example, by guiding cell differentiation, an example would be to install a functional vascular network in an in vitro tissue that would then assist in long-term cultivation. Besides that, I think that there's a general need for in vitro tissues to gain new powers. For example, to improve a quantitative analysis, employing sophisticated biosensors, but also towards shaping the future of tissue engineering towards more explorative applications, maybe organoid intelligence, when we might in the future use neuronal tissues for biological computing, or towards transplantable therapeutic tissues, besides, of course, the relevance of in vitro tissues for basic research. So a bit of background. I originally got in contact with optogenetic technologies as a PhD student. That was together with Professor Wilfried Weber. And at the time, we were working on light-responsive biomaterials that can be interfaced with cells and provide extracellular signals. A short example is illustrated down here, what we call a optotrap biomaterial. It essentially consists of a polymer that is furnished with proteins, in this case the photoreceptor phytochrome B from Arabidopsis thaliana, that can be loaded with various cargo molecules that are equipped with a specific interaction partner, the short PIF tag. And once assembled, we can now use optical signals, the light of a specific color, in this case 740 nanometer far red light, in order to release these cargo molecules into the environment on command. And in a hydrogel, for example, we were able to quite locally and also repetitively release an embedded GFP cargo molecule over a few cycles by applying a 740 nanometer laser mask. So again, this is a biomaterial shown here, so there are actually no cells involved, but we use the same concept and also to control the release of antibodies and then also growth factors to act on human cells. We also attempted to make computing biomaterials, such as the example shown here, a material that would be able to count a number of light pulses before releasing an embedded output molecule. And conceptually for this, we used an AND gate that connects to a time delay and a memory device. It would work as follows. A first light pulse, so here light pulse number 1, would activate the time delay that then in a period after the light pulse would slowly process the connected memory device. And once the memory is activated, it can, together with the perception of light pulse number 2, trigger the AND gate and generate the output, resulting in counting to the number of 2. So we realized this then on a material scaffold by basically embedding an output molecule in a material scaffold via 2 bonds. So there's one light cleavable bond and a protease cleavable bond. And the mechanism works in a way that only after the administration of light pulse number 2, both of these bonds break to release the output, whereas with the first light pulse, one of the bonds still remains intact. So then we can measure that output release is highest after light pulse number 2, and we then subsequently apply the system to control the in vitro enzyme availability of 2 enzymes involved in the carotenoid synthesis assay. Yeah, but then we also moved into cells, inside cells, and here we wanted to control the protein localization in a cell between the cytosol and the nucleus, employing the same photoreceptor. And the idea was here to send proteins from the cytosol into the nucleus using one light color, that is 660 nanometer red light, and then with another light color, 740 nanometer light, let them shuttle back into the cytosol. And we can do that for a couple of cycles. And at that point, we also realized that optogenetics is actually a way to link computers to cells by having them send and sense optical signals. And we try to demonstrate this here by having software running on a smartphone that basically produces an image and projects it to a cell culture. And in the exposed cells, it would then trigger the nuclear import of a synthetic transcription factor in order to generate gene expression patterns. And while that worked, you can clearly see that the resolution that we obtained here is rather macroscopic, and there's also quite some cells among that do not respond, probably due to the transient transfection method that we used here to bring DNA inside the cells. So we're now aiming to engineer optogenetic tissues. I believe it is essential that we stably genome integrate all the required transgenes. That is essential in order to render these systems compatible with long-term cultivation, and also a condition that's indispensable to reflect the cellular complexity of in vitro models. And further, I think that optogenetic gene switches are most versatile in this area. So gene switches, they essentially control promoters, so gene transcription, and here in response to light. And the versatility resides in the fact that the optogenetic components, so here the optogenetic promoter, is separated from the effector. That means we can quite simply swap the effector target gene for another one without needing to re-engineer the optogenetic machinery. And with such stably integrated gene switches, we could then maybe realize many of the desirable features that we would like to have in synthetic tissue models. So we set out and implemented and tested a range of optogenetic gene switches based on different photoreceptors that we find in nature that respond either to red or blue light. And along the way, we learned that while some systems that appear reliable with transient transfection, as also often reported in the literature, they do not necessarily remain functional once moved into a stable context. I mean, this process also goes along with a dramatic reduction in DNA copy numbers, could be one of the reasons. And in the following, I just want to focus on one of these here that we have successfully used quite a bit, which is a derivative of the Avena sativa LOV2 photoreceptor domain that has been engineered into a protein heterodimerization tag, a light switchable protein heterodimerization system originally published by Strickland and colleagues. So in this setup, our gene switch is now activated by blue light. And the mechanism involves the recruitment of a PDZ domain here that is genetically fused to a transcriptional activator to the promoter site, and this happens under blue light. And here at the promoter, this is where the engineered LOF domain resides, and it's tethered to the DNA using a DNA binding protein. And for that, we use the erythromycin repressor protein from E. coli. And that means that we can still provide the antibiotic erythromycin to the system, which then essentially leads to release of the transcription factor from the DNA. And with this, the system is switched off regardless of the illumination condition. And that's a feature actually quite handy to have in engineering stable cell line. It takes time. And during this time, we won't need to fear accidental activation, for example, doing cell monitoring and cell sorting. So we took all the required transgenes together with some selection markers on 2 vectors. So this is a 2 plasmid system. We flanked them by ITRs that are then meant for a sequential genomic and random genomic integration using transposases into mammalian cells, so that subsequently we can perform a clonal selection in order to identify well-performing clones. And shown here as an example, so these are just 24 randomly isolated clones that produce a reporter in response to blue light. And what you can see is that most of the clones can actually produce an optogenetic response. And they also show kinetic and dose response characteristics one would probably expect for a gene expression system, and that we can also use in order to tune the expression strength. So for application and further testing, we then first looked into 2 programmed cell death pathways, namely apoptosis and necroptosis, because these are also good test cases, because you can imagine when controlling cell death pathways optogenetically, we really need all of the cells to respond, because otherwise non-responsive cells that survive the stimulation, they would in the long term take over the culture. So as for apoptosis, you might know that the extrinsic apoptotic pathway engages caspases. There are initiator caspases and executioner caspases, both of which exist in the cells as inactive cymogenes. So they still require further upstream activation. So when we now want to induce apoptosis using a gene switch technology, it would be of no use to express caspases because they already exist. But what we can do is to generate a constitutively active variant of a caspase by reorientating the domain order, so placing a fragment P12 from the C terminus to the N terminus, and additionally installing caspase-specific cleavage sites. And when we express this construct now with blue light, it will fold into a constitutively active conformation, and it's termed a reversed caspase. And then for the necroptosis pathway, we use a different effector, a derivative of the mixed lineage kinase domain-like protein, MLKL, that basically causes membrane rupture. So here are now some mammalian CHOK1 cells that have the optogenetic apoptosis gene switch stably integrated, as it can be seen by the constitutive GFP marker that we have available here. And the cells are doing fine when they grow in darkness. But when we grow them under blue light instead, you can see that all of the cells form very characteristic apoptotic bodies that goes along with a loss of the GFP fluorescence signal, indicating progressive apoptosis. Now, one may ask whether the observed cell death that we see here could be due to phototoxic effect of the blue light treatment. So we need to have a control for this. On the right side here, we now use the photomask to spatially restrict the induction of cell death. That is shown in the upper image. In the lower image, we did exactly the same experiment, but added erythromycin to the culture medium. And to remind you, erythromycin acts here as a chemical shield. It leads to the release of the transcription factor from the DNA. And as we do not see any cell death here, we can conclude that erythromycin is efficient in doing so, but also that the light that we shine on the cells is not phototoxic and does not cause cell death. So photomasks are suitable to demonstrate spatial selectivity, but they are actually not so great for selecting individual cells or tissue features, and they're also mainly incompatible with microscopes. And that's why our collaborator, Professor Mustafa Kamash, and his student at the time, Sam Kumar at ETH Zurich, they have developed a illumination hardware called micro pattern scope that essentially allows microscopic monitoring, and patterned activation of optogenetic systems with a microscope. Essentially, this is a digital mirror device, similar like the key components that we find in multimedia projectors, namely an array of many small micro mirrors that can individually be tilted. And the tilt angle of each of the mirror, together with a connected optical engine, now determines whether incident light is either reflected away from the microscope objective, that is when the mirror is in the off state, or towards it when the mirror is in the on state, so that we can now project light patterns into the focus plane of the microscope. The device itself can be assembled from mainly off-the-shelf components for around about $12,000. Details can be found in our publication. And with that, we can now project light patterns onto cell cultures easily exceeding single-cell resolution. So here we are now at the microscope with a sample of mammalian TOK1 cells that have the optogenetic necroptosis gene switch installed. And on the right side, you see the pattern that we project on the cells. You can also see that we can apply gradients. Down here is an intensity gradient, and the intensity values of the rectangles here, they also have different values. And as I'm going to play the video, you will see that the pattern is also dynamic. So new elements will be appearing here in the empty area. And what we expect to see now is the induction of transgenes in the illuminated areas. So the exposed cells here, they should first become orange due to the co-expression of mCherry, and afterwards necroptotic due to the necroptosis effector, which we can visualize by adding a cell death stain, cytox blue, so the cells will essentially turn blue once they are necroptotic. So I play the video. You see that orange cells appear. New elements are being added that appear a bit later here, and then gradually the orange cells turn blue, indicating that they are now in fact necroptotic. And just to make it complete, the matching control experiment, similar thing, but we added erythromycin to protect the cells from inducing necroptosis. And again, despite the illumination, we see no cell death, indicating that the light pattern that we apply here and the intensity that we use is not phototoxic to the cells. So what we now have available is a extended optogenetic hardware that will either play a role in microscopic monitoring, but also in optogenetic regulation. And now importantly, both of those features can be controlled by software. So software can, for example, operate a microscope, such as navigate positions, acquire images, but then also perform image analysis. So on the right side here is shown a bright field image of some cells, and the image is converted into a binary mask that then can be used to back project the pattern on precisely as a blue light pattern on top of the cells. That means the pattern and the illumination conditions can dynamically change as an outcome of the current microscopic observation, which results in a closed loop, which is also termed cybergenetics. We heard about this before. So to demonstrate this, we implemented a cybergenetic closed feedback by programming a conventional tic-tac-toe game that is executed on a 2D cell culture of optogenetic apoptosis cells. So here we have the software that basically acts as a game handler, and it creates two virtual computer players. First, there's computer player A, who projects circle blue light patterns on top of the cell culture to generate circle apoptosis patterns. And then on the other hand, we have computer player B, who does the same thing, but he plays cross patterns by projecting cross pattern images. So now the two computer players can take turn, scan the position, and decide where to place their pattern. And naturally, as the computer is smart enough not to make any mistake, the game will result in a tie, so there is no winner, unless we artificially sort of tune the parameters that determine the skill levels of the individual players, so that it can happen that at one point one of the player makes a mistake, which in this case would then allow the other player to place three patterns in a row, such as here in the case for player A in the diagonal line. So in this case, player A has won the match. So that's a fun experiment to perform, but more than that, I think it demonstrates a unique combination of stably optogenetic gene switches and an extended computer interfaced optogenetic hardware, and it leaves, as we think, significant implications for the future of tissue engineering and cell signaling research. So the next logical step for us was then to move into 3D, and we asked whether we can also induce genetic events locally in a 3D culture by delivering precise light signals. And for this, we attempted to induce now necroptosis in a three-dimensional tissue. And for that, we generated spheroids and used now a blue light laser of a confocal microscope to induce necroptosis. The sample here on the left is again the control, where we added erythromycin, where we see that despite the exposure here, we do not see an evident cluster of cell death that again would become visible by a blue stain due to the cytox added. However, if we do the same experiment here without erythromycin, we see that about after 4 hours, we have this blue patch of necroptotic cells appearing, this around the illumination spot. Yeah, another application would be to impact Wnt cell communication across three-dimensional tissues. So Wnt is a short distance diffusible extracellular signaling mediator, a morphogen that plays an important role in cell differentiation and doing embryonic development. And when perceived by its receptor, the canonical pathway leads to the activation of a family of TCF transcription factors, and with this regulated gene expression. So what we did here in this experiment is that we generated cells that produce and secrete Wnt, and we also stained them red for identification purpose by having them additionally express a constitutive red fluorescent protein. And when combined in an assembloid chimeric tissue together with another tissue that can sense the secreted Wnt signals and produce a corresponding GFP output, we can now sense and monitor Wnt secretion and perception, spatially and time resolved, as shown here, where a GFP gradient emerges at the tissue interface in the blue light illuminated sample. And a more complex scenario is illustrated down here with a tissue that now holds multiple of these Wnt organizing centers. And due to the spatial selectivity of the DMD, we can now activate tissue features as needed. For example, only one of these two organizing centers. So currently we are working on moving beyond the random integration approach that we use, because that can be problematic, especially when working with stem cells, where the process can accidentally disrupt essential genes. So a much better approach would be to deliver the genes into a safe harbor locus, maybe using targeted CRISPR knock-ins. And to move towards this direction, we first needed to combine all the elements that we need on a single fragment. And before that, remember, we had a 2-plasmid system. So while doing this, we also optimized in a combinational assay the individual elements so that we ended up with a single fragment that we term blue genes that now paves the way for CRISPR safe harbor lock-ins, but it also shows an improved expression, and that potentially extends the range of applications. One example that benefits from an improved expression strength deals with the shaping of tissues, for example, by influencing cell-cell contacts. Cadherins are important here in this area, and by impacting the expression of cadherins on the cell surface, we can, for example, influence the inclination of cells to form three-dimensional tissues, as shown here, where we generated spheroids from illuminated cells that were engineered for optogenetic cadherin expression that grew significantly larger compared to spheroids formed from dark incubated cells. And another idea I just briefly want to mention here is the combination of two spatial methods. One is bioprinting and the other one optogenetic, imaging forming tissues by first layering cells using a bioprinter, and then the next step using optogenetics to guide their differentiation and formation maturation into tissues or to study spatial features, as demonstrated here with plant tissues from our review article. But one exact problem that we actually have, or limitation that we have with blue cheese together with the micropattern scope, is that we are essentially restricted to a single sample because the cells require more or less constant activation. And one way to overcome this is now to utilize bioprinting, which allows us to place multiple different samples in a relatively small area, such as 3 × 3 millimeters, representing the field of view under the microscope. And the example here, we now use 3 individual print heads of a Beyer printer to print down 2 concentrations of suspension cells and also spheroid samples, which then allowed us to assess in a live imaging experiment using the micropattern scope device. So finally, I would like to address a key challenge, which is the integration into safe harbor loci. So the problem here is that the blue gene fragment that we have now here, also depending on the target gene that we use, it easily exceeds 10,000 kbs, and that unfortunately makes CRISPR-HTRs inefficient. And one approach to overcome this is to use the straight-in approach that was originally published by Blanche Arsenio and colleagues. And the idea here is a two-step process. First, we use CRISPR-HTR into a specific locus, such the AAV-S1 safe harbor, to basically install an ATTP phage attachment site together with some selection markers serving as a landing pad. And once we have this one in, we can then use the BXB1 phage integrase to deliver a larger payload comprising the blue genes fragment, and this then enables efficient integration of large payloads, basically without the size limitation. Example, just very briefly, of cells that we generated that way that express a constitutively active variant of the small GTPase Rho A. And that results here in these cells in a cell rounding phenotype that is light intensity dependent due to a remodeling of the cytoskeleton. Yeah, but then a vision that we have is now to use the system for disease modeling. And the idea here is to start with healthy stem cells, or iPSCs, and then genetically equip them with tools, including, of course, optogenetic technologies, so that we have an engineered iPSC from which we can then derive in vitro tissues and then induce the production of disease factors, or maybe the occurrence of disease-associated mutations as these 3D tissues form, which would then allow us to study disease spread and interaction with healthy cells and provide insights that range from basic research to drug and therapy development. However, requirement for this is to have long-term active optogenetic gene switches functional in iPSCs, something we are working on currently, at least with preliminary success. Yeah, and with this, I would like to conclude my talk. Our current research on synthetic model biosystems is meant to advance biomedical research, where it hopefully will impact areas including disease modeling and cell signaling studies, besides other biomedical areas. However, this requires interdisciplinary actions and, of course, the development of tools and methods in order to advance the experimental capabilities of synthetic model biosystems so that they can meet future demands. And with this, I thank you for listening. And before I finish, I would like to acknowledge people who made this work possible. First of all, the current and some former members of my team. Then other people from University of Düsseldorf, importantly, Professor Matthias Zubriggen, who also generously hosts me and my team in his institute. So very thankful for that. And then an incomplete list here of further collaborators, some of which I have mentioned during the talk. And I'm happy to take your questions.
Thank you so much. I mean, you make me smile because you show the tic-tac talk. Actually, when I was a graduate student and postdoc, I wanted to implement, I call it Pac-Man, but now Pac-Man, using optogenetics and then using that tool to navigate the bacteria chemotaxis or phototaxis or whatever. But I
Photon
never
sensing, yeah.
implemented. But you kind of show that kind of thing. I mean, so I guess the Jose had some question or comment. I guess please
Yeah.
or
So
comment.
first of all, a very beautiful talk. Congratulations.
Thank
You have
you.
very nice work. I should add that I know many of your collaborators, Matthias, Mustafa, Wilfred. Next time you
So,
see them, send my regards, please. I always enjoy seeing them whenever we get a chance to cross the big ocean. So say hi for me.
okay.
Yeah, so I have some questions actually. One of them is you mentioned you tried many different optogenetic systems with your melissas. I think it was slide 7 or something around there.
Yeah.
And uh, it's a very interesting list of options that you listed there, but you stuck with the ASLF2. And I'm wondering if you can tell me a little bit about what happened with the others. Did some of them totally fail, or did some of them work, but just
Yeah.
not as good as ASLF2?
Yeah, so this is an ongoing, long-term ongoing thing that we continue ever since, because of each of them, if it does not work in the first attempt, it does not mean it wouldn't work. You need to reconfigure it eventually. Maybe one of the reason for ASLOF2, because you have noticed that we are doing, attempting to do live imaging. So that means you have them on the microscope, and every hour, or I don't know what interval, we take images. And of course, it's important that when you take the images, it doesn't activate the optogenetic systems.
Right.
That's actually a challenge that you first need to think about how to do this. I want to take a picture after 2 hours, but then all cells will be activated because we even take bright field images. So then it contains basically the full spectrum of light. And also we image in the samples that I've showed you, we image GFP, which you actually excite with blue light. So how do we make sure that the other cells are not activated?
Right.
And that's the one advantage of the Arena Sativa LOF2 domain, is the very rapid dark reversion kinetics. Yeah? So you activate it, but then within a few seconds you remove the light, it goes back to the ground state. That means for a gene switch, we need really sustained illumination. If you have just a pulse of a few milliseconds, like acquiring a GFP image, it doesn't produce, it's not productive gene expression. It needs sustained illumination. Yeah, so that's one advantage of the AF2 domain, whereas other factors such as the phytochrome systems, they are very sensitive. They have other advantages that they have two wavelengths, right? You force them back, so you can work with the second wavelengths and 740 nanometer light. But they are bigger and more complicated, and also they require chromophore. So we also have data in some of the manuscripts using the 5B system that also works, but generally it's in our hands weaker, so much weaker than the AS LOF2 system. And then certainly it also depends on the cells that you're using. Yeah.
So should I get from this slide that you tested all these 8 different systems and chose ASLF2 because it was the best?
Well, we still have others that we are still using and for certain applications. So it's not that we have stopped with them, but these are the ones that we have in-house, so to speak. Yeah.
Okay.
And now I talked about our work with the ASLF2 domain. Yeah. I mean, conceptually, others like EL22T2, they're in principle better because they are simpler. We only need one component.
Right.
GAFPO is another one that is here basically a derivative of Vivid. So we have one component that makes things better in principle because we have simpler architecture, and for difficult cells, it's always better to have smaller proteins and fewer proteins. And while EL222, probably you know that works relatively well, I believe, in yeast,
Yeah.
and not so in mammalian cells, to our experience and also the experience of others, even though recently there are publications that they have improved it by exchanging the activation domain. But in the stable context, I think that's still a different story. So
Right.
even though conceptually it's more promising, we are not there yet to have it working in a stable cell line context. Yeah.
Right. So that's actually related to my other question that I had, because you mentioned something about transient transfection versus stable cell
Yeah,
lines.
yeah.
Understood correctly, you're saying these optogenetic systems don't work very well in the stable cell lines? Did I
So
hear correctly?
the thing is, when I started, we used transient transfection. Yeah, and when you look into the earlier publications of, let's say, gene expression systems, they're mainly characterized for transient transfection. So in transient transfection with mammalian cells, you transfect the cells, but only a few cells will take the DNA up. But
Right,
when the cell takes DNA up, it takes a lot of DNA. I mean, thousands of copies probably.
right,
Yeah? In the stable context, it looks different. You can perform the selection, so at the end you have 100% eventually positive culture, but the copy number per cell is very limited. Yeah. So when you use a CRISPR integration, you have 1, 2 copies, and with a transposase, it reaches maybe up to the level of 10, 20. But that's still completely different as if you had thousands or hundreds of thousands of copies in
but
one cell.
in the images that you showed, though, it
Yes.
looked like all of them were responsive. So you showed images of stable cell lines, and you
Exactly. So that's our requirement for if we want to make tissues like here, we need them stable because
right,
otherwise the transient transfection also has the drawback that after a few cell divisions, the plasmids have diluted out. So they're not long-term stable. And we need them long-term stable so that we can have cultures
right.
that we can derive tissues that we can culture for weeks and even months maybe.
So you clearly overcame that limitation.
Yeah. So, and that for us, it's quite some, it takes, of course, much more time and energy to always derive stable tissues and cells. But I think for us, this is a requirement.
Yeah.
Yeah.
So another question for when you showed this tissue, right? This tissue model, what kind of cells were they? I missed that. I missed it.
In which do you mean the three-dimensional
These
here?
organoids, yeah.
This one? So
Kind
this is not an organoid, this is a spheroid. Spheroid, they are made now from the same cells that we used in the 2D experiment here, where we induce necroptosis, but we culture them in a way that they do not adhere to a substrate, like a plastic, but they adhere to each other, forming a three-dimensional sphere.
Are
Yeah.
those CHO cells or HEK cells?
Um, these are CHO cells, but we also have other cells. So this one are, for example, HEK cells. Yeah.
Those HEK cells and
These are HEK cells, and these are CHO cells.
CHO cells.
I
I
should
see.
have specified this here somewhere. Yeah.
Okay.
Here it says HEK cell, but the other one, the necroptosis cells are CHO cells.
Yeah, very beautiful. So actually,
Yeah.
I don't want to take over the entire conversation, but
Mhm.
I would love
Enjoy talking
to,
to you.
offline, have a
Sure.
follow-up chat to talk about your work specifically on CHO cells. I'd be
Thank you.
interested.
Yeah, we can do that.
Yeah.
Yeah, that's wonderful. Do you have any other question or comment, Jose?
Well, I just see the enormous potential of this technology, right? I can
Sure.
imagine that, 100 different ways that you could apply these tools to interrogate basic biology of these cells and tissues, but also, like as you say, list potential therapeutic strategies, right? Like you can induce different
Yeah.
with this exquisite spatial and temporal control on the tissues or the spheroids, to try to understand how the rest of the cells are responding to whatever you're
Mhm.
inducing.
Yeah. And I think the one potential comes when you work with iPSCs that you can make from individuals or from a patient. And at the
Yeah,
same time, you have reproducible engineering methods that are not based on random, but they are reproducible because you engineer one cell exactly the same way as the other, for
right.
example, by utilizing a safe harbor locus. And then I think it could really advance the field.
So actually, another question: Did you or have you tried to introduce orthogonal wavelengths, so blue light and red light, for example, just to control two different set of genes?
Um,
Tried that?
yes, we in transient, yes.
Does it work well?
In I can't really say at the moment, but we have done just now last week was a combination with a chemical system.
I see.
Yeah, so I have one gene on the tetracycline induction and another one with optogenetic, and of course we also have light and transient in also in the work of Matthias and Wilfried. You will see publication where they do this.
Yeah.
by combining different optoswitches, and you have to just to see the crosstalk of wavelengths. Eventually, it only allows you a sequential induction in like one first, then the other, but in other example also orthogonal. But the data that they are published, they are with transient transvection. You don't really know whether the same cell produces both, or is there one cell producing one and one the other, because the data is also not based on microscopy.
Yeah.
Yeah, but it's something also we of course want to use, provided that we have the matching systems working in a stable context.
Yeah. I bet you get this question all the time, but I'm gonna ask it anyways.
Mhm. Sure.
Can you comment about the toxicity that you observe you
Yeah.
induce with light, and how do you control for that? And also, is the toxicity dependent to some level to whether you have the optogenetic component there or not? In other words, toxicity of the light by itself versus toxicity of the activated ASLOV2.
Yeah.
Have you?
Okay, yeah, we had this in discussion. I cannot tell the difference, or if there is a difference between whether or not the cells contain then flavin-dependent optoswitch, if that's your question. But I can say that for blue light, there is phototoxicity, or there can be phototoxicity depending on the intensity. Much less for red light. Red light, it doesn't matter. But blue light, yes, you have to know the threshold, but it also depends on the cells, and it also depends on the environment of cells. For example, for now here, these X cells that we have here, they are sensitive to blue light, for example, when they're in suspension, but they are much more tolerant to blue light when they have adhered to plastic. Yeah. But then there's a threshold. The LOF2 domain unfortunately requires quite some light.
Right.
So in relatively high intensity, but we are fine. We can titrate this out. But then there are other cells like stem cells we are working with. We have observed that they are more sensitive towards light, but it also strongly again depends on their density, like how many neighbors they have.
Yeah. So it'd be interesting for you to look at because we've seen phototoxicity in yeast,
Mm.
but we've seen it usually only when when we're also expressing the light responsive protein, for example, EL222. So
Can be.
just the wild type cell is not phototoxic, but is not photosensitive. But then when you add the EL222, just by having it there, it becomes photosensitive, and that's obvious, right? Because it produces these free radicals as a side
Yes.
product.
Yeah, there are also tools that actually utilize this purpose, like the mini
Exactly.
SOG, right?
Exactly, just to kill cells just by
Yeah.
that side product of the photoreceptor. Absolutely. Yeah.
Yeah, of
Yeah,
course.
I don't have any more questions. Only, a question to follow up later.
Yeah, sure, we can do that.
Yeah.
Sure. Now I see the 10 minutes after, 11 Central Time, but I want to ask one brief question, but I
Yes,
also
please.
want to have one brief answer actually from both of you. So, I mean, you make me so excited because I'm thinking about this one, but I never implemented, as I mentioned earlier. But one thing I really like, especially the Jose's last slide, cyber, kind of idea. I mean, I kind of thinking about the future, and then to get there, I mean, kind of controlling cell, even talking about artificial intelligence related kind of, potential goal. I mean, there should be many challenges you also touch, including some toxicity from the light. In one sentence, what we should overcome to get there? Just one sentence answer from both of you.
Um, it's a tough question to answer in just one
Sure.
sentence, but I can say a couple of things.
Uh,
One of them is that it's not as out of reach as it seems. This technology keeps surprising me every time.
Absolutely.
So one thing to keep in mind is that you do not need so much light. Some of our cells are activated by, to give you an idea, it's stimulating them very briefly with low intensity, and I'm talking
Mhm,
briefly like 1 second out of 100 or 1 second out of 1,000.
mhm,
So it doesn't take that much. And what we need to overcome, I think it's for the control
mhm,
process, I think is going to be a combination of like traditional control theory, but
mhm,
also machine learning, as you were saying, you know, artificial intelligence to try to develop new controllers for how to essentially how to control the cell's behavior. And so I think understanding how to process this data to achieve those controls, I think is the thing that we need to overcome.
Wonderful, thank you. How about Hannes?
Yeah, for now mammalian cells and tissues, I think there's still quite some work to do, in particular also to make other labs adapting to the technology that maybe not really familiar with this. And Jose has already mentioned in his talk that you can purchase a panel for $30, and that's great. But then the final experiment that maybe the PIs in other labs normally have, they will not be able to conduct with a simple panel, but then they want to have spatial resolution and I don't know what. So there, from the technology, there must be made steps forward to make this technology and illumination devices accessible and reliable to the people. But then also, I think the work like what we do to generate cells that really function in a stable context, so that you have all the cells, regardless which one you choose, to respond.
Wonderful. So because of time, we probably need to close now. Okay, let me close. Thank you all for joining and staying today. We'll meet again on October 9, Thursday, the same time, the same Zoom link. We'll have Professor Leos at University College London and Professor Jita Nassari at the Humboldt University of Berlin, Germany. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you. I will stop recording just 1 second. Stop.